This is Jan Dammerholt, President of MyoPain Seminars, with this second part of the module Peripheral and Central Sensitization. Quite early in the development of the construct of central sensitization, people became aware that the induction of central sensitization was dependent on NMDA receptors and substance P. So in this lecture we are going to look at several chemicals that play a role in central sensitization. Substance P, which is co-released with glutamate by unmyelated peptidergic nociceptors, is involved in the generation of central sensitization, as you will see in just a minute. Substance P binds to the neurokinin 1 receptor, which is a G protein receptor that initiates second messenger activity. It causes long-lasting membrane depolarization and it contributes to temporal summation, or increased activity over time. And the time may extend beyond the time of the actual noxious stimulation. Sensitization affects the receptors. For example, the AMPA channel is normally not responsive to brief noxious input. However, continued noxious input causes the AMPA channel to open. With prolonged noxious input, substance P and glutamate are released together, which triggers the activation of the NMDA channel, which next allows calcium to enter the cell. At that point, new AMPA receptors are synthesized and embedded in the cell membrane. But these new AMPA receptors are responsive to brief noxious input, so they accelerate and amplify the response. The process of AMPA and NMDA activation is explained further in this brief video clip.
AMPA and kainate receptors are relatively simple. They require only glutamate binding to open the ion channel, which leads to membrane depolarization and fast excitatory neurotransmission. Activating NMDA receptors is more complex. Both glutamate and glycine, each of which bind to different sites, are required. Moreover, in resting NMDA receptors, the ion channel is blocked by magnesium. Membrane depolarization by activation of a nearby AMPA kainate receptor displaces the magnesium cork, allowing the NMDA ion channel to open. Calcium influx through NMDA receptors contributes to long-term potentiation, a phenomenon that may be involved in long-term learning, synaptogenesis, and other neuronal functions.
As you saw in the video, the essential step is ultimately the activation of the NMDA receptor, which allows ionized calcium to flow into the cell. You can see that the NMDA receptor normally is blocked by a magnesium molecule that is removed in the process of phosphorylation. Activation of the peripheral nociceptive or nerve endings in the muscle causes glutamate, substance P, and calcitonin gene-related peptide to be released at the synapse, thereby activating the nociceptive cell surface receptors. The end result is an inflow of ionized calcium into the nociceptive cell, which initiates the intercellular pathways that maintain central sensitization. Not surprisingly, the pharmaceutical industry is very interested in figuring out how to inhibit central sensitization in the treatment of people with chronic pain. By blocking the NMDA receptor through methadone, dextromethorphan, memantine, amantadine, or ketamine, the later of which is now available as a nasal inhalant for outpatient use, it is potentially possible to stop or reduce central sensitization and therefore chronic pain. However, these pharmacological agents are always limited by their side effects. Before we continue the module, we provide you with a brief list of some acronyms with their actual meaning. I would recommend to keep this short list next to your lecture when you go through the module, and refer back to it every now and then. You do not need to know the full names of all these chemicals, so AMPA receptor or NMDA receptor is sufficient. You do not have to know it by the name of N-methyl deaspartate receptor, for example. Some of them are a little bit more obvious, substance P, SP, that's I think you will be able to remember, but some of the others are really more meant to explain briefly the many complex steps in the process of central sensitization. In this model of central sensitization triggers, you see a synapse between the central terminal of a nociceptor and a lamina 1 neuron under controlled basal conditions. Metabotropic or G-protein coupled glutamate receptors sit at the extremities at the synapse and are linked to the endoplasmic reticulum. You also see the NMDA receptors, and they are blocked by the magnesium, which is shown as a black dot in the middle of the channel. When there is nociceptive input after barrage of activity in a nociceptor, many processes happen at the same time. For example, the primary efferent presynaptic terminal releases glutamate that binds to the AMPA receptor and to the NMDA receptor, now without the magnesium plug, and also to the metabotropic G-protein coupled glutamate receptor. But there's also substance P, calcitonin gene-related peptide, brain-derived neurofactors, which bind respectively to the NK1, to the CGRP1, and to the tropomyosin receptor kinase B receptors. B2 receptors, those are bradykinin receptors, are also activated by spinally produced bradykinin. Nitric oxide is produced by several cell types in the spinal cord and can act presynaptically and postsynaptically. So in other words, it becomes quite complicated. Here's another diagram that shows the complex events that occur at the synapse. The afferent in-coma axonal nerve endings on the left of the cartoon show the release of neurotransmitter substances, such as brain-derived neurotrophic factor, the excitatory amino acids glutamate and aspartate, and substance P. The right-hand side of the cartoon shows the nociceptive cell surface with its cell surface receptors, including the tropomyosin receptor kinase, TRKB, for brain-derived neurotrophic factor, NMDA, AMPA, and the metabotropic glutamate receptor, which are all activated by glutamate. And also you see the NK1 receptor for substance P. Inflow of ionized calcium through the NMDA receptor is shown. This activates the second message systems within the nociceptive neuron that operate through protein kinase A and C, nitric oxide synthase, and COX-2 activating prostaglandins amplify and maintain central sensitization. Here you see a model of a nociceptive dorsal horn neuron synapse under controlled, non-activated conditions. You'll see in the next slide that many of these receptors become sources of calcium. So after nociceptive input, activation of the NMDA receptor and the metabotropic glutamate receptor results in a rapid increase of calcium that activates the prokinase C and the calmodulin dependent protein kinase 2 receptor, which are two major factors of central sensitization. Get ready for the explanation of this slide, because it gets very interesting and complicated. Key intracellular pathways contributing to the generation of central sensitization are depicted in this slide. NMDA receptor activation causes an activation of the protein kinase PKC and extracellular signal-related kinases ERK, the black arrows. Glutamate R1 containing AMPR receptors also activate the protein kinase C, which is indicated by the red arrow. NK1 and CGRP1 receptors also activate the PKC and pKa, the protein kinases, and the extracellular signal-related kinases ERK, which is indicated the orange and brown arrows respectively. The tropomyosin receptor kinase, or TRKB, activates also the protein kinase C and extracellular regulated kinases ERK, indicated by the purple arrows. And the glutamate metabotropic receptors via release of calcium from microsomal stores also activates the protein kinase C and extracellular signal-regulated kinases, and indicated by the gray arrows. So it should be clear that most of the triggers of central sensitization through activation of the NMDR receptor, the mGlu-R receptor, TRKB receptor, the NK1, CGRP, or B2 converge to activate extracellular signal-related kinases, ERK. It is actually even much more complicated than that, because if you just take a look at the AMPA receptor, they are composed of 4 types of subunits. Now you don't have to know all these, but phosphorylation of the AMPR receptors can regulate channel localizations, conductance, and open probability. One of those subunits, the GLU-R1, has 4 known phosphorylation sites, and many others have even more. So it gets very complicated, and it's not an easy process to understand. We clearly do not anticipate that you will remember all this, nor is it necessary. But to show you the complexity, each of these chemicals plays distinct roles that activate or affect other chemicals in the big chemical soup, if you want. CGRP, for example, or calcitonin gene-related peptide, potentiates the action of substance P and brain-derived neurotrophic factor. But it also activates the second messenger pathways protein kinase A and C. It was released in the skin in migraine, although its role in the skin in migraine really is not known. So second messengers are molecules that relay signals from the receptors on the cell surface to target molecules inside the cell, in the cytoplasm or in the nucleus. Let's take a look at a few other of the chemicals that play a role in central sensitization. If you take a look at bradykinin, which is an inflammatory kinin that is induced by intense noxious stimulus, it acts through the secondary messenger system with the protein kinase A and C and the extracellular signal-related kinases. And the result is that it boosts synaptic activity. Bradykinin is actually a chemical that tends to make things worse. Under normal circumstances, we only have B2 receptors, and those are not really activated by inflammation or by persistent nociceptive input. However, when there is inflammation or when there is persistent nociceptive input, the membrane starts making so-called B1 receptors. So that's a neuroplastic change, it's an adaptation, and these B1 receptors are very responsive to nociceptive input, which in effect makes the problem for the patient worse. So this is an example where the receptor responsiveness is very dynamic because it changes from a B2 to a B1 receptor. As I stated before, in normal muscle tissue, the B2 receptor is much more prevalent. But with tissue inflammation or with persistent nociceptive input, an additional bradykinin receptor is synthesized in the cell body of the ending in the dorsal root ganglion and inserted into the nociceptive terminal membrane. And this B1 receptor is much more sensitive to nociceptive input or inflammatory input. And as if that is not bad enough, as I said, the B1 receptor is inducible and involved in sensitization of the peripheral nociceptor, but induction and binding of the B1 receptor can also lead, and that's problematic, lead to the production of pro-inflammatory mediators, including tumor necrosing factor alpha and interleukin 1 beta. All these chemicals have been found in the immediate vicinity of active myofascial trigger points. And just to reiterate this point, stimulation of B2 receptors, the receptors that we have under non-inflammatory, non-noxious stimulating circumstances, leads only to transient increase in the intracellular calcium concentration. And for that reason, sensitization of the nociceptor is really not that likely. But once B1 receptors have been synthesized, stimulation of these B1 receptors results in prolonged elevation of intracellular calcium concentrations, which can lead to sustained peripheral sensitization. And where it gets very frustrating is that even after the inflammation subsides, if the source of peripheral nociceptive input is eliminated, the conformational change of the BK receptors may persist. In other words, the receptors did not get the message that they no longer have to allow calcium to enter the cell, and this may actually change the transition from acute to chronic pain. So this is actually a problem where the chemicals start a process that may be biologically functional, but once the input changes, for example, the inflammation subsides or the nociceptive input is removed, the process may continue, and now you have a patient with chronic pain or persistent pain who has no true objective findings that explain that pain. Another chemical that can cause a lot of trouble is serotonin. Serotonin is significantly increased in chronic trapezius myalgia, for example. The action depends a little bit on the cell type and the type of receptor it targets, but serotonin activates many pathways that contribute to central sensitization, such as the intracellular signaling pathway ERK or extracellular signal-related kinase. And as soon as you get activation of all these systems, the problem tends to get worse rather than better. So in a way, it's questionable why the body creates these chemical interactions that make the problem of central pain and central sensitization worse. As you probably already expected, there are many receptor classes and subclasses of serotonin. 5-HTR receptor is the most common one, but there is also 5-HT1, 2, and 7, which are also linked to myalgia. In conclusion, available microdialysis studies of chronic myalgia indicate that serotonin is involved in chronic muscle pain. So where does serotonin come from? In the periphery, it is released from platelets and mast cells, often related to tissue damage, but they can sensitize the afferent nerve fibers. Serotonin can activate the extracellular signaling response kinase pathways, and it's activated by intense noxious stimuli, but it can amplify and maintain central sensitization. In this case, by a means that occurs as a result of the action of activated cell surface receptors, particularly AMPA and NMDA, which activate the extracellular signaling response kinase pathways. So it's a post-translational mechanism. As I just said, the extracellular signaling response kinase pathways maintain central sensitization. It boosts the NMDA receptor activity, it increases the NMPA cell membrane numbers, and it decreases potassium channel activity, and that increases membrane excitability. Since the body does not seem to be able to correct these chemical mechanisms, the pharmaceutical industry is very interested in creating drugs that enhance descending inhibition. The drugs that are commonly used are so-called serotonin norepinephrine reuptake inhibitors, such as duloxetine, which is perhaps the best studied of the 4 drugs shown here. These are also known as antidepressants. These drugs enhance descending inhibition, especially in chronic pain, by increasing the activity of the supraspinal, so centers in the brain, that mediate descending inhibition, so as the ventral rostral medulla or the periaqueductal gray matter, for that as an example. In addition to bradykinin, glutamate, and serotonin, there are many other chemicals that all play a significant role in initiating and maintaining central sensitization. So just when you think you got a grip on it, we are going to introduce something different, namely spinal cord glia, which in this article are sometimes called the new players in pain. Glia cells historically have been thought of as supporting cells of the brain, supporting structures for neurons. But glia cells are what causes neurons to become overactive inside the brain and spinal cord. It's the glia cells that make neurons supersensitive to pain, and they perpetuate the pain message. So where glia cells traditionally were found to be nothing but supporting cells, glia cells actually drive the process of central sensitization. So we'll spend a few minutes on glia cells because many schools of physical therapy, and medical schools for that matter, do not really study glia cells and provide very outdated information. Glia cells cause this disruption in normal nerve function by releasing inflammatory mediators that enhance pain. So glia cells are actually very busy cells that play a very active role in creating central pain states. The model we have used so far in these home study modules is actually a very outdated model because it does not consider the role of glia cells. It is now known as a classical pain signaling model, as you see depicted here. So when a painful stimulus is encountered, such as stepping on a tag as in this example, peripheral pain responsive nerves, the A and C fibers, are excited. These axons transmit action potentials to their presynaptic terminals in the spinal cord dorsal horn. If the signal is strong enough, neurotransmitters released bind to and activate postsynaptic receptors on pain transmission neurons, white dynamic neurons, which in turn ascend contralaterally through the spinal thalamic tract to the brain, carrying the signal to higher centers, giving us an experience of pain. So under normal circumstances, when we have maybe minor aches and pains, glia cells are present, but they really are not doing much. So nociceptive signals, or maybe so call it quote pain signals, arriving from the periphery along the A and C fibers, cause release of substance P and excitatory amino acids in amounts appropriate to the duration and intensity of the pain stimulus. Activation of the NK1 receptors by substance P, activation of the AMPA receptors by these excitatory amino acids causes the transient depolarization of the pain neurons, pain transmission neurons. They were generating action potentials that are related to the higher pain signals. We've already discussed that. The NMDA receptor under normal circumstances does not play a role in minor pain signals because it's plugged by magnesium, as we already saw earlier. In the classical pathological pain model, glia cells are not being considered as creating a pathological role in pain perception. Because in the classical view, which we have already reviewed in detail, once the substance P and glutamate are released, eventually in the pathological pain state, the magnesium plug is removed, and that causes lots of problems as we have discussed. But as you see in this diagram, it also causes the conversion of L-arginine to nitric oxide. Nitric oxide is a problem gas because it rapidly diffuses out of the neuron and acts presynaptically to cause an exaggerated release of substance P, and postsynaptically it causes the neurons to become hyperexcitable. Again, in this classical pain model, glia cells are not being considered, which is in fact a mistake. When we do consider the role of glia cells, you'll see that it gets much more complicated. But there is now a lot of research that shows that glia cells are in fact a significant driving force for creating and maintaining these pathological pain states. So the role of glia is superimposed on NMDA and nitric oxide driven neuronal changes we just discussed. But glia cells are activated by three main sources. First, by bacteria and viruses that can bind specific activation receptors expressed by mitoglial astrocytes. Secondly, substance P, excitatory inflammatory acids, and ATP released by either A or C fiber presynaptic terminals or by brain to spinal cord enhancement pathways, which are not shown here, can be a factor. And thirdly, nitric oxide, prostaglandins, and fractalkines released from these terminal neurons, pain terminal neurons. Following activation, these mitochondria cells and astrocytes cause a hyperexcitability and an exaggerated release of substance P and excitatory amino acids from the presynaptic terminals. These changes are created by glial activation of the p38 mitogen activated protein kinase pathways and the release of nitric oxide, reactive oxide species ROS, excitatory amino acids, prostaglandins, and pro-inflammatory cytokines such as interleukin 1, 6, tumor necrosis factor, and nerve growth factor. So it gets very complicated, but again, many of these chemicals are found also in the active milieu, immediate milieu of active myofascial trigger points. Again, we do not expect you to remember all these details, but you should know that glia cells are the driving force behind these neuronal pain issues that we have discussed. Glia cells can also play an integral role in developing tolerance to opioid pain medications, thereby blocking their effectiveness. So sometimes if people don't understand why opioids no longer work, it could be because of glia cells. If you're interested in looking at glia cells in more detail, we highly recommend the literature by Dr. Linda Watkins, who has published quite a few articles on glia cell activation, and those articles are actually quite understandable. It's intriguing to know that opioids like morphine bind to receptors on glia cells. But instead of causing them to quiet down, the morphine can actually activate glia cells to go into so-called pain mode, so that they release more and more pro-inflammatory mediators, which makes the pain get worse, as opposed to get better. So glia cells can be one of the reasons why opioids are not always working. So there's a lot of new research on trying to figure out how to control these glia cells. We hope that these lectures made it clear to you that understanding of central sensitization is critical in figuring out what to do in manual therapy, whether it's manual trigger point release, joint mobilizations, or dry needling. Manual therapists can apply the science of nociceptive and pain neurophysiology to the practice of manual therapy. Clearly, the diagnosis of central sensitization in an individual patient's musculoskeletal pain is not straightforward, and you many times need an interdisciplinary team to really get to the bottom of it. The outcome of the diagnostic process can be used to determine the appropriate treatment parameters, such as the intensity, frequency of various manual therapy techniques. The article by Jonijs, van Boudenhoven, and Oostendorp on the recognition of central sensitizations in patient musculoskeletal pain is a very good article of the application of pain neurophysiology in manual therapy practice. As you can see from this chart, many medical diagnoses of chronic pain, such as chronic whiplash, fibromyalgia, irritable bowel syndrome, are characterized by central sensitization. On the same token, many other chronic pain conditions have central sensitization present, at least in some subgroups. Some people with low back pain, temporomandibular disorders, myofascial pain syndrome, osteoarthritis, rheumatoid arthritis, etc. And common signs and symptoms of central sensitization are hypersensitivity to bright light, hypersensitive to touch, noise, pesticides, mechanical pressure, medications, temperature, and maybe even electromagnetic fields. Symptoms that may be related to central sensitization are fatigue, sleep disturbances, unrefreshing sleep, difficulties with concentrating, a swelling feeling in the limbs, tingling and numbness. So recognizing central sensitization is the key in manual physical therapy practice and other disciplines. While this may look like a commercial of an advertisement, our new book, Manual Therapy for Musculoskeletal Pain Syndromes, edited by Cesar Fernandez de la Peñas, Joshua Cleland, and Jan Dammerholt, emphasizes the new role of pain sciences in myofascial and musculoskeletal pain syndromes. From the foreword written by Rob Oostendorp, I'd like to read the following. Manual physical therapists should be familiar not only with biopsychosocial context of pain, but also with modern insights derived from pain neuroscience and psychology concerning reconceptualization of pain. In fact, a sustained biomedical approach can even lead to an iatrogenic effect, which results in an increase in pain. Although increasing evidence supports a role for psychological and social factors in the emergence and persistence of chronic musculoskeletal pain, the majority of clinicians received an education with a solely biomedical focus, a focus that is also evident in the profession of manual physical therapy. This focus is reflected in a long tradition of treatment options for patients with musculoskeletal disorders based on biomechanical principles. This emphasis on biomedical aspects appears to shape a therapist's knowledge, attitude, beliefs, and behavior towards chronic musculoskeletal pain. An additional barrier to the acceptance of this emerging biopsychosocial model is that the emergence of new or revised theory and subsequent changes in practice are often characterized by a significant time lag. And with this book we try to make a contribution to the transition from manual therapy in a narrow sense to manual therapy in a very broad sense, including the application of modern neurophysiological and psychological insight. And we hope that with this book we created a resource that will not only benefit patients, but also manual physical therapists, body workers, chiropractors, etc. worldwide. And that brings us to the end of the lectures about peripheral and central sensitization. I'd like to emphasize that you really do not have to memorize everything from this last lecture. This is rather complex materials. If you understand some of the changes that happens with serotonin, bradykinin, substance P glutamate, NMDA receptors, AMPA receptors, that is more than sufficient. It is important to become a mechanism-based clinician who considers the latest evidence in clinical practice. Thank you so much.